Yes, so this will be, I promise you, the last talk. So for those of you who are sick of me, but I will try to continue on what we have discussed yesterday and a little before that, and I will try to see how the molecular properties of motors, which to some extent are learnt by the trying to understand the mechanical cycle of those motors, trying to understand the biophysics of those motors, of individual motors, how their single molecule biophysical properties may be important for understanding collective transport inside cells in a more complicated situation. Now, this is something that we have attempted to do in a few situations. It is not always possible to do this, so please take it with a pinch of salt. There are interpretations and there are controversies, and I will try to discuss them, and I would like to hear your feedback on these aspects. Just a very brief recap. This may not be necessary. We are going to look at the right. So we are essentially looking at. Okay. We are looking at objects like these, which are transporting cargoes. And one of the important points is that there is a lipid membrane on this cargo, which is not often thought of when you try to devise solutions to this problem. And these are some numbers which we have discussed yesterday, and I will not go to them. They are written here. You can read. And the big question is that if you have a cellular structure like this with such a messy arrangement of microtubules, it can be a little different in different situations, and other kind of proteins and filaments which are not shown in this picture. How do you, in a sort of deterministic way, localize different things to different places inside the cell? How do you generate polarized distributions? Why is it not a soup inside? Okay. How do you generate polarized distribution of cargoes, which is in turn necessary for so many kinds of biological downstream cellular processes and indeed physiological processes, as we have discussed on the first day in one context. Right, so these motors, you can think of them as undergoing this kind of a transition between two potential energy landscapes, and this is what I was trying to draw yesterday. And you diffuse once you are here, and you have some chance of going to the next well, and then once you go into the next well, much less chance of going back. So this is sort of a one-dimensional. You can progress in a particular direction along the microtubule or actin as long as such a potential surface exists, potential energy surface exists. Now, as we also earlier discussed, typically that is not what is going on inside in the real world. You rarely have one motor of a very precisely defined type carrying cargoes. It is the more likely situation is that you have several motors collaborating together in ways which we do not understand to transport cargoes. And so perhaps this is a more realistic depiction of of a cargo inside a cell, where you have different kinds of motors that are generating force in different directions, functional on this. Some of them may be attached, some of them may not be attached, some of them may be on the other side and may not be able to reach the microtubule. So given this kind of a situation, they are interrelated, but you can try to address each of these questions separately also. So what are the kind of forces that they are generating at the level of a single motor? And if you take 3 of these green motors, do they generate thrice the amount of force? That is not necessarily true always, okay? Sometimes it may be true. What are the numbers of the motors? Now, when I say numbers, I can mean the numbers which are present in a biochemical sense on the entire cargo. Maybe that number is 20, 30 of a particular motor type, we don't know. So that is one kind of a number. The other kind of a number is, of that 30, what is the fraction which is proximal to the microtubule so that it may attach? of those that are proximal to the microtubule, a certain number may actually have attached and, and be able to generate force. What is the compliance of these motors? The compliance of this link, that is important to understand in terms of the tenacity with which these motors might work. Also important is the compliance possibly of this structure, which includes a lipid membrane and perhaps a liquid filled environment inside. So that also has a compliance. How does that matter? The fluidity of this membrane, in other words, how does that matter for this entire, entire process? How is it controlled? I mean, for biology, it's really important to understand. This is not a static picture. This can change, and I will show you one example. Then also, something that has been less discussed, I have just randomly put these motors at whatever places, wherever I wanted to put them on the membrane of this cargo. That need not be true. These motors, the red ones versus the green ones, or maybe there are other colors also, they might be organized in a specific manner on the lipid membrane, and that may be, again, be important to deciding how they will work together or not work together. Now, when you look, when I came into the field and started to look at these problems, what has been done over the years, there's a lot of literature on the kind of, here is a motor which is attached to a cargo. What are all the proteins between, so this is the business end of the motor, this is the dynein motor, and there are literally tens or hundreds of proteins that are recognized in linking this motor to this cargo, okay? And each of them has its own function. They are absolutely very important in perhaps determining this regulation on a certain time scale. But is it, I mean, coming from a, from outside a biology background, my view was a little bit like this, okay? Do I really need to understand all of these, accessory proteins to, get anywhere? And the reason we know about so many proteins, because the biochemical techniques to isolate motors and the adapters, these techniques are very well developed, and therefore, we have a very large literature on this aspect, okay? Now, perhaps one possibility is that the time scale may be important. The temporal scale on which these factors play a role may be important. And again, I can't make a general statement, but it is quite possible that if you want to understand how these are functioning on a shorter time scale, over shorter time scales, maybe seconds or tens of seconds, the biophysical properties of these motors, if you can measure them, may be able to tell you more than what each of these does. Over longer time scales, particularly in relation to cellular signals, maybe these will become important as well, and you have a slow regulation. Again, this is not a general statement, but this is one thought which one can consider. Okay, all right. Now, there was a paper, and I don't think, Stephen, you're there, hi. So, yeah, so in 2008, there was this very influential paper from Dr. Klump, Lipovsky, and their team, where they talked about the possibility of what I was trying to kind of allude to in the previous slide, that so when you, okay, let me go back. The implication of so many proteins sitting in between this and this is that all of these somehow regulate the motors, and that makes sense completely, okay? Now, not only does, let's say, a protein X sitting here, there are known proteins which bind to both kinds of motors, the green and the red ones, okay? And they might bind in a mutually exclusive manner in response to a phosphorylation or addition of some ion. So it is quite possible that there is a traffic cop. That's what has been shown here. Think of these proteins as traffic cops who will give the red signal to one motor and the green signal to another motor. Okay, so there's a regulatory complex which sits on top and tell you go and you go. And this kind of a picture has been very, a lot of people have talked about this, and there are evidences pro, for, and against this. But what in this paper, again, I'm not going to go into the details of this, but what they considered a situation where there are dynamic transitions. So there are two kinds of motors. Just think, I mean, the names are not important. Let us assume that there are two kinds of motors, and one, the blue motor wants to go in one direction and the yellow motor wants to go in the other way. And if we think of stochastic transitions between these motors, where each has its on rate and off rate and other biophysical properties, which I'm not getting into right now, then what happens as a result of that? What does this cargo do effectively? If these are going on and off and transitioning between these possible states, then what they showed using computer simulations is that you can have stochastic transitions between these two species of motors, leading to a back and forth motion. Now, why is this bidirectional motion important? Because as Sandhya also showed you and many others have seen, that back and forth motion, or which is called bidirectional, where you go in one way and then stop for a while and go the other way and then again continue, that has been noted for many kinds of cargoes for a long time, okay? And therefore, there is a significant effort to try and understand the most simplistic picture in which such kind of bidirectional transport might happen. And perhaps this paper argued, and Dr. Klum can correct me or add to this, that there is no need for a third complicated coordination complex, at least on perhaps some time scales. Of course, these proteins are sitting there, attached to the motors, and they do something, yes, but then I believe that the time scale on which you are looking at this becomes important. Yes, Kavita. Yes, you can. Yes, absolutely you can. Yes, I will show you an example of that. Yes, yeah. All right, so I keep going back to the computer because I'm not used to this. And in a later paper, what they also, something that they showed, and this is really interesting to me, is that if you have this kind of a model, which I showed you here, and develop that further, and you plot, you give certain biophysical properties to the kinesins and you give certain biophysical properties to the dyneins, and I'm not going to get into the details of that, but if I make a phase diagram, okay, where, what are these different colors? These different colors are where you can have only plus directed motion. That's what you were asking. I will come to more experimental demonstration of this later, or a mix of plus and minus, where you bidirectional motion, or all kinds of possibilities, and a situation where you have a state where you pause for some time. Zero is where you are not moving or barely moving, depending on what your experimental resolution is. And so this kind of a phase diagram was developed, and there were further developments, improvements to this kind of a hypothesis, where people considered, and Ambarish actually is not here today, but some of his work also looks at situations when these, let's say there are n number of motors on this, and they may not be sharing the load equally, okay? They're trying to transport something against opposition, and whether they share load equally or not is a question and a controversy, and they may or may not be sharing load equally. What is the force velocity response? I will come to that a little later, and how long the data. So these are developments which have happened later, but I won't be able to go into too much details of this right now. Following on, and taking some inspiration from your work, what we did is that we tried to address this issue in a more in an experimental fashion. This is when, right, I had started my lab and I didn't know what to do, honestly, and we stumbled upon a system which was very nice, and we took it further. What we did is that we took dictyostelium cells. Okay, these are amoeba that grow in the soil, and you can purify endosomes from the dictyostelium cells. And luckily, given the biochemical conditions that we have and all that, you take the endosomes out of the cells, and you now put those endosomes on a microtubule which you have stuck to a cover slip, and you can see very nice, robust motion of these endosomes. Okay, and again, by using the kind of techniques that I discussed yesterday, optical tweezers and related biochemistry and tracking of these cargoes, what we concluded eventually is that, and this is one, I think I can't run the video from here. So you can see, yeah, so when this runs, you will see that there are two membranous objects linked by some kind of a membranous tether. And if you look at this, your first, you will see, I feel that this is certainly a tug of war situation where one is pulling and the other detaches, and then the other is pulling and the other detaches. Okay. Now this you can see in where two things are joined together by a long membranous structure here, or even if I'm not showing you the video for that, if you have a single cargo like this, that also moves in a back and forth manner. Okay. So this is a system where you can reproduce the back and forth motion of cargoes on a cover slip, and we can track these cargoes moving around, and we on this cover slip on the microtubule, and we can track them moving around inside cells as well. And within reasonable limitations, we are fairly sure that the motion outside cells is very representative of what happens inside cells. Okay. Now, right, okay. So what we concluded, again, I'm not going to go into the details, is on an individual cargo like this, you often have a situation where there is a single kinesin motor. This is the Ank 104 kinesin, which exerts a force of about 5 to 6 piconewtons. And that motor, single motor, is opposed by a larger number of dyneins, which are weaker, each of them. Okay. And so, but because there are a larger number, they're approximately force balanced. Okay. And now, in this kind of a situation, this team of opposing motors, they may somehow be able to coordinate or not coordinate. But the result of this, that you have motion where you move in one direction, stop for a while, don't do something, then go back, and this happens. Now, again, this is not just a biophysical curiosity. This motion, saltatory, bidirectional, back and forth, or whatever you want to call it, is really important to biology. Okay? Why is it important? Because when cells take in foreign material from outside, maybe nutrients, maybe pathogens or other kinds of organelles, I will come to that later, there is an initial period where the endocytosed or the phagocytosed material is transported in a back and forth manner along microtubules. And this back and forth transport is really important for the properties of that membrane on the endosome and the proteins and the lipids to undergo very programmed maturation, programmed changes. That is so because as this object is moving along these microtubules, there are other objects also, other kinds of cargoes, which are also moving along the same microtubules. And it is very likely that if you move around, rather than when you are just sitting at one place, you will make friends, right? You will meet some other cargo, you will exchange membranes, and you will exchange proteins. So this is a very well-established field. I'm not going to go into that, but this saltatory motion is very likely to allow the maturation of these endosomes, leading to their final transition to late endosome and probably a lysosomal fate. And this is also important for, I will come to that a little later, for the clearance of many pathogens, okay, which when they are ingested into cells. Right. So to try and address this, what we try to develop a system where we grew cells of different kinds. These cells could be dictostelium cells, which I was telling you about. Yes, Sandhya. We have done that experiment not inside cells. So I can't give you the last part of the answer, does it end up in lysosomes? Okay, so, but it is known that dynein inhibition will block lysosomal fusion. That's not our work, that others have done. Yes, yeah. It's hard to answer. Correct. Correct. Correct.
And is it about controlling the rates of maturation, which is much more important?
Yeah, I don't know the answer to kinesin. I know the answer to dynein. I don't know the answer to kinesin. I should look it up. Yeah. Right. So what you can grow cells. So these cells can be either these amoeba, the dictyostelium that I was telling you about, or you can take mammalian cells which like to eat things. Okay, those are cells of your immune system, of your innate immune system, which are macrophage-like cells, which will phagocytose solid objects, and we feed these cells with plastic beads. Okay, 1 micron or smaller, and so this is a macrophage cell, and the nucleus is somewhere here or here, I forgot, on one side. And once these beads get in, you can, this is real time. This is not sped up or anything. You can see that they, move quite vigorously. And what you can do is that you can pulse these. So the nice thing about this system, there are several nice things according to me. One is that you can add these beads to these cells at a defined time point, allow them to mature for a known duration of time, and they will undergo changes. Basically whatever happens to them inside the cell, they will undergo maturation progress, and you can block that maturation simply by taking it down to 4 degrees centigrade, making it cold, and then you can burst open the cells and purify these beads either at an early stage or at a late stage, or perhaps at intermediate stages, which we have not done yet. Okay. Now, the nice thing is that these beads, they have their density is lower than whatever is around them. Okay. So you can then take them, depending on the kind of bead, of course, it's not a generic statement, but the density is different from the rest of the stuff. So you can put them in some sucrose, and you can take them to a centrifuge and spin them, and you can separate them from the rest of the stuff in the cell. Okay. And then you can store them in small, you can take 5 microliter aliquots, put them in a buffer and keep them in the freezer and come back after 10 days. 10 days is okay? No, 10 days is not okay, within 5-6 days. And you can, what you can now do is that you can add some ATP to it. You can now put them in a cover slip with the microtubules, and they will move if you have done everything right. Okay. So these are phagosomes, phagocytosed particles, and right. So essentially what we believe we are able to do is that rather than look at a system where you have a very artificial plastic bead to which you have attached a motor, you don't know exactly how you have attached it either, and we can transition from this to something like this, okay, where you have the plastic bead, yes, that is allowing you to see these objects, to purify them, and the nice thing is that they are of a precisely known size and highly refractile, so you can use a laser to trap it, okay, and generate a very known calibrated force. But in addition to all these advantages, you have a lipid membrane covering it, and you have motors which have attached to this in an in situ manner, in situ meaning that the way it naturally happens inside cells. And so you have something like this, okay. Now you can take this object and put it on cover slip and see what happens. Now, this is one thing that we did. We also did measurements where we directly investigated the motion of these things inside cells by shining a laser like this. So something is moving, you shine a laser at the right time and you trap it inside the cell, and you do these kind of optical tweezer measurements. We did that also, and I will briefly show you that. Now, there are significant challenges to doing these measurements. These have been done before us using lipid droplets inside the embryo of the fruit fly by the lab of Stephen Gross and others, and that is the lab actually I joined for my postdoc, but I then developed this system for my own purpose, okay. Now, so here is a cell like that. Here is the nucleus, and you have these things moving around as I had told you. Now, the problem in these kind of measurements, because this is a sort of a school, so I want to go into a little bit of this, is that when you are trapping, you're using optical tweezers in a in vitro assay, in a cover slip, then you have a microtubule stuck here and you have things floating around, okay? And you catch, your optical trap is coming in from below and you have a focus here, you catch an object which is floating around. So when you do that, that object is automatically centered at the center of the optical trap, okay? And then of course fluctuating around that position, and then you take it down. So more or less, if you do things right, your centering in the optical trap is guaranteed, okay? That is not true when something is moving inside cells, okay? Because the object is already moving and you want to focus your, so suppose something is moving at 1 micron per second velocity, which is a fairly high velocity, okay? And then you, suppose this guy was moving and you want to trap it right here. It is not easy to focus the laser beam on exactly the center of that spot. And if you don't, now the center is in, you have to think of the center in two contexts. One is, suppose this is a microtubule and your laser beam is going to come in. One point is that here is something moving and you should be right at the center of this, okay? So one is center in this aspect, the other is center away from the microtubule. If you focus your laser beam, you can't see the microtubule. If you focus your laser beam here or here, even 30-40 nanometers away, okay, then you will be applying a force away from the microtubule. And then that will, that will create a lot of complications in your measurement, okay? So these problems do not exist if you trap a cargo in solution and bring it down, but these problems are significant when you do this exercise inside cells, and perhaps this was the reason why there were a lot of controversies about these kind of measurements. There still are. I'm not saying there aren't, but yeah. Yes. No, they won't, because there's so many foreign bodies entering all your cells all the time. You drove from somewhere and you inhale diesel fumes. Those are all foreign particles which are going into the, the macrophages in your lungs. Yeah, that's the size of many kind of bacteria.
You never see them rejected?
No, you will, ultimately you will, yes. So they can be, so these are, so the bacteria are processed, right? They're broken down, and that's why, we are alive. Okay, so the bacteria are processed down, their proteins are chopped off. These things can't be chopped up. They will go into the lysosome and they will sit there. So, okay, okay, I should have just shown this. So basically, if your trap is focused right here, when your object is here, you are fine. But if you're focused here or you're focused where, so then there is a problem. And we had a lot of time, we spent a lot of time. We actually took about 7 years to figure out all of these things. And then we could develop the, abilities to do these experiments, we believe, in a better way. And anyway, so those are things that I don't want to go into right now, but what did we learn from this, okay? So now remember that in these cells, we know what is the plus direction, okay? We know where the end of the cell is. So the microtubules are, so this is the nucleus, right, so this is the nucleus. Most of the microtubules are with the plus ends here. We showed that by different assays. Let's not get into that. And the minus ends are here. So if something is moving like this towards outside, you have a good chance that it is kinesin driven. If it is moving inside, then there's a good chance that it is dynein driven. You cannot be 100% sure, but by and large, that's what is going to happen. So then we did these kind of measurements where we trapped these objects. We had the advantage that we could exert a significant force because these beads are highly refractile compared to what is around them, and that is obvious because you can see them clearly. Otherwise, you would not be able to see them, okay? And we could trap these, and we could now measure these kind of stalls, which I have talked about yesterday, so I won't explain that again. And we now measured the force, and we know that this particular, we did some biochemical assays to figure out that the motor involved is the conventional kinesin that exerts about 6 piconewtons of force. So we saw events like this, okay? Now the interesting thing here was that, okay, let me come to that later. And then we made a histogram of how many events are driven by 1 versus 2 kinesins based on this information, if that interpretation is correct. And we think that largely about 90% events of these phagosomes are driven by 1 kinesin. That does not mean that there is 1 kinesin on the phagosome. That's the 1 kinesin that is able to engage at a time and generate force, and sometimes there are 2 kinesins. We could also purify these phagosomes from these cells, and look at their transport in vitro, and we see pretty much the same kind of. So remember, these are done inside cells. These are, we have pulled out the phagosome and then put it on a cover slip and then done it in vitro, okay? Right. Now the surprise, so this was kind of expected, okay? And this was expected. So one interesting point was, if you look at this duration, you remember in the earlier slide I talked about tenacity. How long can the given motor work if I am pulling it back, okay? So the tenacity, this duration for which you stay above, so let's say this is the 10 piconewton is the maximum force that you can generate, and if I say that the duration that I can survive over half maximal force, that is 5 piconewton in this case, I will take that as a measure of tenacity, okay? And then I measure that, I call it T stall. Think of it as tenacity, the duration of the stall, and you will see that the T stall duration is not, here is not much longer than this, okay? That was one interesting finding. I will come back to that. Okay, let me go on and I will come back. Now, when we did the same measurement for the cargoes that were moving in the other way, towards the minus sign, towards the nucleus, we saw something very interesting, okay? That depending on the magnitude of the force, so the magnitude of the force tells you roughly how many motors are working. And the duration of the force tells you how tenaciously, how they are working, or can they cooperate to work. If 5 motors can cooperate, you can expect their function to improve, okay? So when we looked at dynein in vitro, now this, oh sorry, I forgot, I should have a scale bar here, I have somehow chopped it off. But so this is essentially, this is time, okay, along this axis, and this duration is about a second. You can see that as the force generated increases, or in other words, as the number of motors who are working is larger, the time for which they survive against half maximal load keeps on increasing, okay? And this is quite dramatically seen when you make a plot like this, okay? And so what I'm plotting on this axis is the stall force, and what I'm plotting on this axis is this T stall, this thing, okay? And for dynein, you see that it is more or less a straight line, okay? So as you increase the number of motors, the capacity to survive against load increases almost linearly within this range, okay? But when you do the same thing for kinesin, that is not true. 2 kinesins, 3 kinesins survive pretty much for the same time as 1. And this was really interesting because perhaps the dynein motor has an ability, this means that the dynein motor has an ability to work in large teams, okay, which kinesin cannot. And this kind of agrees with in vitro experiments on kinesin, which had been done around that time, and people had shown that if you take single versus 2 kinesins, there is, it doesn't work better. There's negative interference between 2 kinesins if they're trying, 2 or more kinesins if they're trying to work. All right. Okay, so so kinesin 1 certainly, kinesin 2 also, this is known. I don't know about the other ones. It's hard to purify all of those, and many of them are not even processive, so this doesn't apply. Yeah. So this is true for Ank 104 because we have done it in, it's a dicty version, but we have not done in the mammalian one. Yeah. Yeah. So are there any other questions, or I will move on in that case? So then the question arises that if dynein can work in large teams, yes. We can't, experimentally we can't go beyond this. Sorry.
Last 2 points.
You mean these two? I mean, so there is theoretical work which has been done which suggests that there may be a tendency to saturation, but I mean, I really can't say anything on the basis of this. It's hard to get a lot of data at this range. I don't know, really. At some point it will saturate. I mean, at some point you will not even have the geometry where more motors can attach. So it's a question which at least biologically may be irrelevant. Okay, but maybe there are situations. So I don't know the answer to that. So then the question is, why is one motor? What is there inside the motor? What is there at the single molecule level which may allow it to work in a large team, dynein, whereas the other one cannot? And what some of my work had shown when I was doing a postdoc is that so this is the head of dynein. So this is how this is an animation of how dynein moves around, and so this is the AAA head of dynein, and this is the stalk by which it attaches to the microtubule and walks. And there are multiple ATP binding sites within each head, as you see here, 1, 2, 3, 4, and some of them can hydrolyze ATP, some of them can hydrolyze but cannot bind, some of them hydrolyze weakly, and this kind of a situation. So this guy is much more complicated than kinesin, which can bind only one ATP in one head at a time. Okay, this can bind more. And what I had shown in working with Steve Gross is that is that this ability to bind multiple ATPs allows the dynein motor to function as a gear. Okay, what do I mean by that is that it can, when you pull it back, and then what is likely happening is that you bind different numbers of ATPs depending on the load, if ATP is available. And when you bind different numbers of ATPs, you can exhibit different step sizes. Okay, and the importance is that the work done per cycle remains constant. So when the step size is higher, the distance is higher because you're taking a larger step, but the force is proportionately lower. Okay, when the step size is lower, you can generate a higher force. So the product of the force into distance is more or less constant, and that is the work done per cycle. And that is exactly what happens in your car when you use your clutch to change your gear and you want to go uphill. Then you go slow, but you can generate more force to go uphill. So then if all that is, if you believe me, if all that is true, then how could this ability to shift gears allow dynein to work in large teams? Okay, and for that, I missed one slide here. Okay, let me draw it. I don't know how I missed it. Okay, so what we measured in that paper, I forgot the slide, is that we measured something called the force velocity curve for these motors. Okay, and the force velocity curve is where you have this optical trap data and you measure this. I mean, you can do it in different ways. I'm telling you how we did it. It's a rather crude way, but you measure the slope of that. So you have position versus time. Remember, so you are getting events like this, and where this is time and this is position. Okay, so you measure the slopes at different places, which gives you a velocity. And when you measure the slope here, you know how much you have stretched your spring, so you know the force. Okay, so you can make a force velocity curve. And when we did that, what we found is that the force velocity curve for dynein was like this, but the force velocity curve for, I mean, I'm not going to all the details, for the force velocity curve for kinesin was a little bit like this. Okay, so this is dynein, this is kinesin. Okay, so this is velocity and this is the load under which it is working. Okay, so now what this means is that at moderate forces, the dynein slows down very rapidly. Okay, but kinesin doesn't do so. It keeps going for a while. Okay, now again, this is a hypothesis. We can't prove this. This is happening at the size scale of very small size scales. But what this implies is that if the leading dynein motor slows down when it sees a small load, then it will wait for the ones behind it to catch up, which are not seeing any load because they are behind. Okay, and this might allow these motors to work to bunch together. So they take steps of different sizes. The one at the the leading motor reduces its step size quickly, okay, and waits, therefore goes slow and waits for the ones to catch up, and then they may bunch together. And based on this kind of force velocity curve, and this might allow these motors to work better in a team. Another property of dynein, which might be very important to this, is the ability of dynein to enter into a so-called catch bond state, okay? And that is something which also I will come towards the later part of my talk, and that's something which Dvitya is also trying to address. So anyway, if you want to read more about it, this these are published work, and you can. I have time until 10:30 and 40. So are there any questions until this stage? Because I will move on to something little different now. No, it may happen, but we have never addressed that specific question. We have never addressed that specific question. So you would have to do really real-time force measurements in the presence of an obstacle. Well, the team size does change in the sense that there are detachments and attachments, so the team size might be fluctuating. Yes, but now coming back to what you asked, maybe I'll give you some kind of an answer now. Okay, not in response to obstacles, but in response to something else. Okay. Say that again. Yeah, it's a mix of dictostelium. Some of these experiments have been done in dictostelium. Some have been done in mammalian cells, mouse macrophage cells. These two kinds of. Some of these experiments have done with motors purified from the brain of goat also. So there's a lot of different kinds of systems. You can't answer all questions in a given system. Yes, absolutely. Yeah, it has been done in mammalian macrophage, not in an animal, but in a cell from an animal. Yes, yeah. Okay. Later, working on the same, what we kind of Ashim and Divya, they made a very interesting finding, okay, when we continued working on this, and which was the following. There's a lot of light. I don't know if you can see this. Remember that I had told you that when we use this model system, we phagocytose beads. We allow these cells to eat beads, okay? And we can, so the beads enter the cells, and we can stop their maturation at a 5-minute time point, within 10 minutes, or at a 1-hour time point, okay? And when we do so, then we block that maturation at early time point or a late time point, and then we now burst the cells open and purify these objects. And so we can purify either early phagosomes, which were stopped within 10 minutes, or late phagosomes, which were stopped after an hour, okay? Then what we did is that, we were not looking for this, but we wanted to see whether dynein is present on the surface of this cargo. Remember that this cargo, it's not a bead anymore, okay? It has a lipid membrane around it, it has all kinds of motors, all kinds of proteins, okay? So we used an antibody against the dynein motor, and we tried to see the distribution of dynein. We actually just tried to see dynein, is it there? We were not so ambitious at that time, okay? But repeatedly what we see is that, so these are 2-micron diameter size beads that we had phagocytosed and taken out, either at an early, EP means early phagosome, LP means late phagosome, okay? So the distribution of dynein on the surface was remarkably different for early versus late phagosomes, okay? You can see that the dynein is more or less uniformly distributed. Here, the dynein appears in this kind of tight puncta. Along with this difference of dynein, what we also saw, I'm not showing you here, is that the amount of cholesterol on the membrane of this phagosome increases when you go from early to the late stage. So these guys have much more cholesterol than these guys, okay? Dynein is known to bind to another protein called ORP1L, which in turn is known to bind to another, RAB protein. And I mean, let's not go into the details, but ORP1L, to which dynein binds, is known to be a sensor of cholesterol. Okay, so one would therefore be led to believe that dynein, directly or indirectly, likes to bind to cholesterol on the membrane in this situation. And because we have more cholesterol here and less, what we proposed is that cholesterol, along with cholesterol and sphingolipids, along with other proteins that are present on the membrane, allow the membrane to reform and form what is called as lipid raft-like domains. Okay, so what we hypothesized, and I'm again not going to go into all the details, you can read more if you want, but that you are starting to form lipid raft domains, nanometer or 100 nanometers of that order size domains on the surface of the phagosome as it matures inside cells and as it acquires more and more cholesterol by interacting with later components of the endosomal pathway, okay? And then eventually you have late phagosomes which have a large amount of cholesterol. And so how does this help, okay? How does having uniform versus more clustered, so we called it clustered, dynein is clustered into the, how does it matter, okay? What we found, again, I'm cutting this short, is that when you are in this state here, you have dynein that is uniformly distributed on the surface, okay? And that's what I've shown you here. But as you go through these cycles of maturation and you acquire cholesterol, you form something like this, okay? And therefore, because if dynein is attaching to the cholesterol, you now start to have large teams of dynein on certain parts. And if this guy is oriented in the proper manner, you will have one or more of these dyneins attaching to the microtubule and driving transport. In this situation, you will normally not have a large number of dyneins attaching because they are all over the place. Okay, you just won't have enough. So quite interestingly, the overall number of dyneins on the surface of this and this is the same. We have characterized that using biochemical techniques. I'm not going to that. But what seems to happen is that the dyneins which are on the surface, you don't add more or remove anything. You just redistribute them on this membrane and start forming these kind of patches. An interesting aspect of this problem, which we have not addressed, we would like to, is that this lipid raft domain on which these are sitting is supposed to be more mechanically rigid. Okay, because it has cholesterol and sphingolipids, it is ordered domain, and because of its mechanical rigidity, you may be able to apply a larger reaction force against it. Okay, and that's why you can assemble such a large team. That's a hypothesis. We don't know. It would be interesting if one could address that. Okay. Yeah. So what happens then? This is in response to your question. Is that in this situation you typically have 3 to 4 of that order dyneins working together because they're all over the place and not enough of them are proximal to the microtubule. In this situation, in contrast, you have a lot of microtubules which get here and you effectively increase the team size, okay? And they can generate large cooperative forces, and let me come to that a little bit. So this is what you have, okay? So imagine a cricket ball which will fit in the palm of your hand, or a football, okay, which is much larger. And these are supposed to be cigarettes, so I'm just trying to scale it to that ratio, okay, depending on the size of the cargo to the motor. So if I take motors which are shown as the cigarettes and put it on a cricket ball, because the motor is roughly the size of the cricket ball, the cargo here, if I have to access this microtubule, I have only if I am on the bottom half of this, that's only when I can access it, okay? Otherwise, most of these, if I am here, I can't even reach the microtubule. But if I am a football, okay, even if I am on the bottom half, very often I won't be able to access it. So what is the solution here? If I want to improve transport for large cargoes, large with respect to the size of the motor is what I mean. And remember that the motor is 100 nanometers, of the order of 100 nanometers. What can I do? And perhaps, so you can simply do, let's say you know the size of this motor, okay, and you can calculate an area. So now what I'm doing is that this is the arc. Given the size of the motor, 70 nanometer, there's a certain arc from which I can reach this, okay. Otherwise, if I'm above this, I can't reach it. So what I'm doing here, I'm taking this object and just turning it by 90 degree and looking at it from the bottom here, okay, and that is the area of this patch, okay. I mean, it's an approximate calculation, but that's the area of the patch, and I can calculate the area of the patch, and then I can calculate if I randomly throw a motor on the surface of this object, then what is the probability that the motor will come to this patch, okay. It's simply the ratio of the two surface areas, all right, and I can calculate that, and this is what I get. So if this is the radius of the cargo, and if this is the probability that I will be able to contact, I will fall into that, I will do bullseye, okay, then as the cargo radius increases, that probability really goes down. In other words, if you have a large cargo, then increasing the number of motors is not a solution, okay. You have to somehow put them into certain domains where there's rotational diffusion going on all the time, okay. So if they are in the right orientation, then if you are proximal, then you will be able to attach, but you have a lot of motors close together. So, so what I was trying to say is that, this is, for a large cargo, this may not work. You might have to do something like this, okay. And now this is an image on a GUV, okay, giant unilamellar vesicle, where this kind of domain-like formation. So this is stain for cholesterol or a related protein. I don't remember exactly what, but you can see that these kind of domains can actually form a lipid membrane. That is well known. Okay. All right. Okay. Yes.
In your experiment, do you actually see that in the early stage there are only small cargos involved, and in the late stage?
No. The cargo size does not change. The cargo size is the same. So the cargo size, because that's the bead, right? So the bead is the same size, 1 micron or 500 nano. That size remains the same. But when I want to go from an early to a late phase, then to improve transport on the same size cargo, I make this kind of patches, is what I'm claiming. Okay. Some later work, this will be relevant to something that I will bring up later, is what Paulomi with, working with Ranjit, what we were able to show is that if you have, again, I don't think I have any slides on there, so I'm not going to go into the details of this. This is also published, you can read about it, is that when you take these early phagosomes, remember the phagosomes which had just come in and I had stopped them, so they move in a bidirectional manner, okay, and I will show you evidence for that. If I take one of those cargos, early phagosomes, and catch it in the optical trap, and keep it on a microtubule, I will see, maybe I will go to that slide, that will make it life easier. I should have put that here. Oh, oops. Okay, I have missed, here, okay. So, yeah, so this, don't worry about the elliptical size, assume it's spherical, so if I take this object and trap it in the optical trap and put it on this microtubule, now there are both kinds of motors active, okay, and therefore then I do the standard optical tweezer assay in a little non-standard way, okay. Here I have time and position like what you see earlier, but now you will have events in both directions. So what I have shown you earlier and what you would have seen typically in the literature, that you see only blips on one side, okay. Here you will not see that. Here you will see that sometimes dynein is active when you are going down. So this is the geometry, okay. So here is the phagosome, the early phagosome, it is held in the optical trap, and this side is the minus end of the microtubule, that side is the plus end. So sometimes you will go this way, sometimes you will go that way, okay. And you now have, you look at these events over for many cargoes, and what you do is that you call these events either K events or D events, kinesin events or dynein events, and you have two kinds of reversals. Two kinds of transitions. One is K followed by D, okay, D followed by K, or K followed by K, or D followed by D. There are 4 possibilities, K or D, so there are, there are 2 possibilities, kinesin or dynein, so there are 4 combinations. Now you just score how many of these event pairs, these are event pairs, right? One event followed by another event. How many event pairs are of the D type? D, sorry, D.
What is the event here? Which one is winning?
No, the event is there a force generation towards the plus end of the microtubule or minus end of the microtubule?
Which one is winning?
I won't say winning, most likely the other one is not even engaged at that time. Okay? So, yeah, so you have a so you have pairs of events KK, DD, DD, KD, and you see that if you just score them, okay, you find that the counts are approximately equal for all kinds of transitions. Okay, so means that once you have done a kinesin event, there is equal probability of doing kinesin or dynein next time. Okay, it's as simple as that. Now that is really interesting because that very likely would guarantee you bidirectional motion, right? Otherwise, if you were in one type and you continue doing the same thing, then you won't have bidirectional motion. So this system, whatever it is, okay, at least from these measurements, seems to give you a bidirectional transport where you go in one direction, don't do something for a while, go back. So now the question is that here is bidirectional motion happening. Does it require me to think of a complicated process or a regulatory process? But can we explain the statistics of this by simply, a tossing of a coin? Okay, where the coin in this case is unbiased. You have D or K, and again, I'm not going to go into the details of this right now. That is what we were, that's what our analysis told us. Whether it happens or not is a different question, that this choice between dynein and kinesin can be simply decided by the tossing of a coin. So you don't need any complicated regulatory mechanisms. However, the regulatory mechanisms, their role might be to bias this coin simply. So there's a hypothetical way of thinking about transport, that you have a coin where you decide this way or that way, but that coin need not be a just your regular coin. It can be a biased coin. That biasing is happening by all those other proteins which are attached to these motors. Okay, so that can happen over different time scales. Now,
Would there be any correlation in the sequence? Which event followed by which one?
we didn't see that. So that's what, yeah, you're talking about the time. I just give you the population scores here, but we don't see that. We don't see that.
So given there is a KD, it is equally likely to be followed by another KK or KD.
Yeah, all possibilities. So rather think of it as simply, if there's a K, it can be followed by a D or a K. That's, for me, it's easier to understand it that way. Okay, then what we have done more recently, here is Paulomi and Pankaj, who have really done a fantastic job looking at this. And the experiments were done pre-pandemic, but all the analysis was done sitting at home during the pandemic. But so that there's a so we are now, so this work, it used early phagosomes because we wanted to see bidirectional transport. What we did later is that we used late phagosomes because we wanted to understand dynein function. Okay, and we know that the late phagosomes move towards the minus end of the microtubule, so they are almost 100% dynein driven. Okay, so we don't want to get into complications of kinesin right now, so we looked at use late phagosomes simply. Okay, and we developed another assay which I'm not going to talk into right now. We developed a system to do lipid dependent recruitment of motors to cargos. Okay, so what we have done in this paper is that we take a bead and we deposit a lipid membrane on it containing different lipids. One particular lipid that is interesting is phosphatidic acid. Okay, when we take such a cargo with phosphatidic acid and add a cell lysate, ATP releasate actually, from that, specifically the dynein motors gets attached to this phosphatidic acid and you get really nice dynein driven motion. So this is a So purifying these motors is complicated, and I'm not a biochemist, so for me to do this routinely is a nightmare. Okay, but here we have developed a really nice system where you can use the lipids simply to select the motors. Okay, so you get really nice minus directed motion, and when we do these kind of experiments, the question that we were trying to address is that is shown nicely. So here are the dynein motors. Remember that there is not one dynein, the red ones are dynein, but there are multiple dynein motors. But what is happening for each dynein is that dynein is walking in this manner, and there is another protein, one of those regulatory proteins that I was telling you about, called dynactin. Dynactin, the interesting thing about dynactin is that one part of dynactin binds to the dynein motor, and the other part of dynein at the other end seems to skate along the mycotubes like this. Okay, it can also bind mycotubes, but little weakly. Dynactin cannot hydrolyze ATP. Okay, it doesn't even bind ATP, but this guy can. Okay, so why is dynactin needed for this? And this is a really important question to understand, because there are mutations in dynactin leading to many kinds of neuropathies. Okay, so what we thought of doing is, now you can think of the dynein dynactin machine as something like this. Okay, that this person is walking with a stick, which is the dynactin. Okay, now if I am able to inhibit this binding versus this binding specifically, okay, I don't want to change anything else. Okay, then I either prevent this person from grandfather from holding the stick, or I prevent the stick from binding to the road. Okay, so that's what we were trying to do in this case. So what we did, so what we did is that we took a small peptide which is the part of dynactin or dynein, depending on what you want to do, basically this interfacial region. Okay, remember, this experiment is being done in a cover slip, okay, where I have purified these cargoes, and I add a small peptide, and if I add lots of that peptide, by mass action, it will go and block the binding of this and this. Okay, so I can selectively inhibit the binding of dynactin to dynein at this end, or I can take a peptide or an antibody against this red part of dynactin and prevent this binding from happening. Okay, and when we do these experiments, I'm not going to this, is you can read about it, is we see very distinct responses. Okay, when we inhibit this binding, we find that this entire machine can get started on the cargo, but it can't sustain its force generation. Okay, but when we inhibit this, it has a starting trouble. It can't even get started. Okay, and so then many of it is interesting because many of the problems with dynactin mutations happen because long distance heavy load, the tenacity of the machine seems to have disappeared. Okay, so perhaps this binding is more relevant in those cases. But again, I won't have the time to continue on that. You can look it up. Yes, Sandhya.
Dynactin binding actually helps dynein walk better or bind.
Which one? You mean this one? Yes, that is known. Absolutely, that is known. Yes, it's not only Trina. Several people have shown that this binding is required to maintain a persistent contact with the mycotube.
Where do you situate this with respect to your teams of dynein which can work cooperatively?
Uh,
I'm thinking that dynactin will have a greater role when dynein works in isolation rather than when it works in teams.
That is possible, but I don't have any actual, because these measurements that we have done were done on a team. Not some of them were done on a single, but did we have? It's an interesting question, but we have not really addressed that. It is possible, what you are saying, that the effect might be stronger on fewer motors compared to a larger team.
The reason I'm asking is because there are few large cargo things, especially in flat cells which are not neurons, you will have large
Correct.
The minute you come to neuron, the cargo
It's
kind
small.
itself is very small.
Yes, yes.
So your ability to organize like this
Is difficult. Yeah, and your curvature is high, so you can't, yeah, so you can't. Yeah, so it is possible that if dynactin becomes more important for motion driven by a few dyneins, then a problem with dynactin will become more important in the neurons.
Exactly.
It is possible, but it's, yeah.
Is a dynactic always associated with a dynein motor?
Most, I mean, the only known functions of dynactin are with dynein. And there, you are asking, is there free dynactin in the cells? Is that?
I'm asking, like, if we repeat this binding between dynactin and dynein, then it is just the cargo with the dynein. So, will the movement be hindered or inhibited in some
It will be different. We know that if you purify only dynein, remove the dynactin, still you do see motion. That is known. The motion is poorer, but you do see it.
Okay, so you mentioned that there will be a starting problem for the dynein if we remove this dynactin linkage.
Correct.
But it will still move, right?
It will move, yes. Once it engages, it will take more time. It will have, more hiccups to engage. But once it engages, then it does fine. Right. So I have till now I've talked about more or less things that are already published, but I will talk about in last few minutes. I will talk about something that we have been trying to do in our lab presently, and this is about. So I will just go. You can. Okay. So that's something that Dvitya is doing with help from some others in the lab. This trying to address this question of dynein's catch bond. Okay. Now, yes, the catch bond is known, and what a catch bond is, I will come to in a minute. But we were very intrigued by the possibility of what happens after a catch bonding event. Okay, that is something that has not been addressed to the best of our knowledge, and let me try to explain. So, what is this catch bond business? Here are stalls of a cargo, a lipid droplet, which is driven by dynein inside this one. This is George Shobeta's work, so it is inside Drosophila embryos, okay, or on purified lipid droplets as well. Now, what happens is that here is the lipid droplet moving, driven by dynein, and this shaded part represents when I switched on the trap. I switched on the trap here. Okay, so after this, you are under load from the optical trap. Before that, you are moving freely. Okay, how long do you survive after the trap is on? This is similar to that T-stall thing that I was telling you about, and you see that for this is for kinesin, okay, and this is for dynein. Okay, you see that for kinesin, you don't. I mean, I'm not going to the detailed statistics of this, but you don't survive for a long time. You detach and you fall down. Okay, but for dynein, you do survive for a while. Okay, and what people have also been able to show, and Ambarish is not here today, but his work also has shown that dynein seems to have this ability to switch into a catch bonded state. What is a catch bonded state? Where, let's say, imagine you have two loose links like this, and there is a hook here. Okay, if I have a hook like this, if I pull it suddenly from the edges, the hook will engage. Okay, and that you can think of crudely as a catch bonded state. But if I don't have a hook, if I have just some weak interactions along the length of that, then it will be slip bond. Okay, then it will not engage. All right. So dynein has this ability to catch bond. Exactly the molecular details of that, what amino acid, all those things are not known. So I'm not getting into those details. Okay. In other words, if I take dynein and put it suddenly under a high load, it goes into this state of a catch bond where it now is tightly holds onto the microtubule for a while. Okay, eventually it will let go, but and that property of dynein may be useful, what I was telling you earlier, in this large T stall where it has this tenacity against load. Now, the interesting thing was, I go back to this plot. Okay, here we found that these reversals happen. Now, for both of these kinds of motors, we know the rough number of motors, the red motors or the green motors, the dyneins or the kinesins, and from data that has accumulated over the years, a lot of people have contributed to this, we know the on and off rates of these motors in response when they're moving freely or when they're moving under a load or under various conditions. Okay, So now the question is, if I have this kind of a situation with multiple dynein motors, maybe 6 or 7, something like that, and 1 or 2 kinesin motors active, and if each is going on and off and has a certain run length, then what happens to this system? Okay? What we believe is, from on the basis of some simulations that Dvitiya has tried, is that in this situation, kinesin would rarely get a chance. Okay, because there are just so many dyneins on this and they're given their on rates. So in other words, if an early endosome comes in and this is the kind of distribution, you would not get bidirectional motion. Okay, we haven't proven this, but just on the basis of simulation. So then the question is, how do you ensure bidirectional motion? Okay, how do you ensure a large number of dyneins so that they can force match the kinesins? And over and beyond that, how do you ensure that you get transitions? How do you give kinesin a chance at all? Okay. So what we hypothesized, therefore, was something called a reactivation delay for dynein. Okay. Now this is completely a hypothesis. We have some experimental evidence for this, but we are trying to work on it. What is the hypothesis? Is that once dynein goes into a catch bonded state, where it is trying to move and suddenly you pull it back, it engages a catch bond. Okay? The catch bond survives for a certain time. It's not going to survive forever. But then you detach from that catch bonded state. You detach from the microtubule and you go into an off pathway state. Okay? Where, because you were under this high load, you have changed your conformation in what way we don't know exactly. Okay? You have gone into a funny off pathway state and you need some time to recover from that state. And during that time, you cannot rebind the microtubule. Okay? So, therefore, and this may be happening to more than one dynein. We have done some simulations, we are trying to address this in an experimentally also, which Ditya will talk about. Once this delay period has expired, this is the time, okay? This is the time where you see that after one dynein event, you take a little time, okay? So what we find is that if you impose a catch bond on dynein, which we have tried to do experimentally, the dynein team takes more time to re-engage, okay, compared to the kinesin team, but this is still work under progress. We will see how it goes. So I think I will stop there, and that this situation where it goes into an inactive state gives kinesin a chance, and that's probably a reason why you see these kind of equal probability of all kinds of events. You get flips in both directions. Otherwise, you would see only dynein events. Okay. Is that my last slide? Yeah. So and I don't want to go into this too much. So we can force a catch bond here onto dynein, we believe, by using a piezoelectric stage, basically pull the carpet from under dynein, okay? And then once you have done that, measure the time until the next dynein event. We are right now building up the statistics for this. We will see how it goes, okay? I don't want to get into this too much more. I will stop here. I would like to thank these agencies at the end. That's a picture of IIT Bombay Powai Lake. The possible positions for postdoctoral fellowships are open in my lab. You people can, if anybody is interested, look into it. This is about a year-old picture of the lab. Mukesh, Pankaj, who are, Mukesh has moved on to do a postdoc at Cornell, and Apurva, Paulomi, Bhavna, Dvitiya is here, Joel, that's me, Shubham, and a few people are missing or have come in later. That's that from me. And I will stop here. If there are questions, we can go.